So this really explains, I think, the vast majority of the false negatives. We simply didn't have a mutation in the right place in the right time, so we can't see the event or the process. Also, of course, the coalescent process itself is a very stochastic process. And so this evolutionary stochasticity, sometimes just by chance, we will generate a pattern that we would interpret one way that would just really arose by chance. And that could give a false positive. Also, selection can be operating on a particular locus, and that can really change the geographical distributions. And we don't know a priori when we look at a given gene whether or not its spatial distributions have been modified by selection. So both selection and evolutionary stochasticity can lead to false positives. And I don't think you can ever really completely eliminate them because you can't eliminate these two sources. By the way, the evolutionary stochasticity, you've got a sample of 1 for any given haplotype tree because the species in its history is never replicated. And that's a big statistical problem that a lot of people don't realize. We can replicate across genes, but we can't replicate the history. Okay, we have one example of it. Okay, and back in 2002, I published a paper in Nature that basically argued that these errors can be minimized by studying multiple loci. So I no longer do single locus nested clade analyses. I think it's very error-prone. What you do is multi-locus nested clade analyses. And after you've analyzed each locus separately, you ask, are the inferences in type, place, and time, can you cross-validate them across loci? Because we don't expect the topologies of the trees to be the same. But if we had a range expansion, for example, we might see the signature of that in many different loci, at least in those loci that happen to have the right mutation at the right time. And if we look at enough loci, we should see that signal across more than one locus. And so that can eliminate a lot of the evolutionary stochasticity, the problem of the false positives, and selection because it acts idiosyncratically on each locus. So doing a multi-locus nested clade analysis is, I think, the best way to avoid both false negatives and false positives. It doesn't completely eliminate them, but it certainly minimizes them. So I did this. This was a data set I assembled in 2005. I decided you can constantly update this because the rate at which genetic data is coming in is amazing. But I figure I'm only going to do it about once every 4 years. Otherwise, I would go crazy. So 2 years ago, this were all the samples on 25 different DNA regions where there was enough sample size across the globe and enough geographical sampling that I could study it. So that, like nested clade analysis is a statistical procedure, so you have minimum requirements in terms of numbers of individuals and also numbers of locations. And although there's a lot of DNA data out there, a lot of it was not gathered in the right way for these kinds of phylogeographic analyses. So this summarizes the 25 regions. It includes, of course, mitochondrial DNA, the Y chromosomal DNA, and then a whole bunch of X-linked and autosomal genes. Each haplotype tree was checked for recombination, which is very important for these groups. And so I'm only dealing with the non-recombinant areas. And moreover, I estimated the trees with statistical parsimony, which quantifies the degree of uncertainty in the tree. And the only inferences that I retain are those that are robust to that uncertainty. So I don't regard my trees as given. I regard them as estimated with error. Now, how do you cross-validate? So, for example, the first thing is concordance of inference type and geographical location are pretty straightforward. So, for example, when we applied the nested clade analysis to these 25 DNA regions, they collectively yielded 15 inferences of range expansion, so that's concordance of type, going out of Africa into Eurasia. So that's concordance of geographical location. So we had 15 inferences that were concordant by type and location. Okay? But we also require them to be concordant in time. So were these 15 separate expansions out of Africa into Eurasia, or one? Okay. That's why we have to come to this. And this is the hard one. These are easy. This is the hard one. So the first thing we do is we need an estimate of time. And so I estimate the time of an event or process by the age of the youngest haplotype or clade that contributes to that inference in a statistically significant fashion. Because that's when all the genetic variation that was marking this event is present. And so we estimated with the molecular clock, but I used some older theory developed by Tajima and Kimura to show that given that currently you have K mutations that have accumulated since this event occurred, this is the probability distribution of the time given the true time and the number of mutations. Okay, and we'll estimate the time from the molecular clock. And in fact, this equation incorporates two sources of error. It incorporates, first of all, the evolutionary stochasticity of the coalescent process, because the variance of this time is proportional to t squared, and that comes from coalescent theory. It's also inversely proportional to the number of mutations that are marking the event, and that comes from the stochasticity of the molecular clock. And it's kind of, as geneticists, we're caught between the rock and the hard place, as we say. Because if we have short times, and therefore the coalescent error is not so great, we usually have few mutations. Okay, so the error goes up. On the other hand, as we get bigger times, we get more mutations, but t squared goes up really fast. So the amount of temporal information you get from any one gene is very limited. Okay, and I think sometimes we tend to overblow our degree of precision in molecularly dating events by not taking into account both sources of error. So it's not just the molecular clock error, the error associated with K, but it's also the coalescent error. So we can now plot these gamma distributions for those 15 inferences of out-of-Africa expansion, and here's what we get. So this shows the loci and the 15 events that occurred. Okay, and we don't care about the variance of these distributions because the variance depends not only on time but the number of mutations, and that's going to vary from locus to locus. So we want to test the null hypothesis that all these gamma distributions have the same time, the same mean, not necessarily the same variance. And we can do that using a maximum likelihood framework. So I developed a likelihood ratio test of the null hypothesis that the estimated times from J loci are all the same, and it's this G statistic. So here we have the number of mutations that were calibrated for the molecular clock for locus I, the time for that locus, and then we have a pooled time under the null hypothesis that they're all homogeneous. And this allows us to test concordance in time. So now we have all the elements necessary to do this cross-validation procedure. And so when we apply this likelihood ratio test to these 15 inferences of out-of-Africa range expansion, we reject the null hypothesis that there was only one expansion with a p-value of 3.89 times 10 to the minus 15. And that's a very small number. So we can be very confident that there was more than one expansion out of Africa in human evolutionary history. Not one, but several. I think you can also, by kind of eyeballing this, see that these distributions are clumped into three major categories. Another thing you can do with the likelihood ratio is you can partition it into statistically independent subsets. And when you do that, you find that there's great homogeneity within each of these three categories, but highly significant differences between them. So this null hypothesis framework says there were at least 3 out of Africa expansion events into Eurasia. Not 1, but 3. And there could have been more, but my training is also in statistics, so I have no evidence for more than 3. There could have been more than 3, but I have no statistical evidence to reject that hypothesis. I can definitely reject the hypothesis of only 1 out of Africa expansion event.